OpenStax, Psychology, 2nd edition, Chapter 3, Biopsychology, narrated by Dr. Nani Asman, University of Hawaii, Maui College, for Psychology 100, Survey of Psychology. Biopsychology has to do with the biological mechanisms that affect or underlie behavior. Among those in this chapter, we're going to talk about genetics. We're going to talk about the nervous system, including neurons and the actual nervous system itself, and then how the nervous system interacts with the endocrine system. Studying human genetics can help researchers understand the biological basis underlying the different behaviors, thoughts, and reactions of humans. Among the questions that often are asked, you have examples like, why do people infected by the same disease have different outcomes? Like today with the coronavirus, right? Two people can both be exposed. One can end up with mild symptoms, and the other person could end up in the hospital on a respirator. Why is that? We also wonder whether there are genetic components to certain psychological disorders, like depression. You know, if someone in your family has depression, does that make it more likely that you will? Which, of course, gets to the next part, how genetic diseases are passed through family lines. We talked about Charles Darwin's theory of evolution a little bit in Chapter 1. That was the example with the, I think, giraffes, either giraffes or dinosaurs, and the trees that lost their lower leaves to some sort of fungus. And so only the taller animals survived. So that just helps us to remember that it isn't supposed to be that it's the smartest or the strongest species, that part of the species that survives. It's the ones that are the most adaptable to change. And so some of those characteristics would be those that help protect against predators, those that increase access to food, and those that help to offspring alive. In case you didn't know, Charles Darwin's theory of evolution by natural selection is in his book On the Origin of Species. If the theory of evolution says that the most adaptable will survive, why do we still have certain genetic diseases that cause people to die, and it hasn't become less common? One example would be sickle cell anemia. This is a genetic condition that tends to afflict people of African descent. And what happens during a sickle cell attack is that the normal round blood cells become these kind of crescent or sickle-shaped blood cells that have pointy edges, which can be very painful during the sickle cell attack, because you can quite literally feel your blood passing through your veins. But also it has caused certain people to die at an early age. One of the reasons why sickle cell is still around is because sickle cell helps people to be immune from malaria, which is a deadly disease carried by mosquitoes in Africa. And so in this case, being able to carry the gene would make you better suited for the environment. Genetic variation has to do with the genetic differences between individuals. These variations contribute to a species' ability to adapt to its environment. It starts when the 23 chromosomes from your mother's egg is fertilized by the 23 chromosomes from your father's sperm. Now, a chromosome is a long strand of genetic information known as DNA. DNA is, of course, deoxyribonucleic acid, which is the helix-shaped molecule made of nucleotide base pairs. In each chromosome, sequences of DNA make up genes. And a gene is a sequence of DNA that controls or partially controls physical characteristics known as traits, such as eye color, hair color, etc. A gene may have multiple possible variations or alleles, which is a specific version of a gene. And so a gene could be coded for hair color or eye color, and depending on the genetic coding it gets from the other parent, that will determine the outcome for the new creature. The difference between genotype and phenotype is that genotype is referring to literally what are in your genes, what's the genetic makeup of the individual, what's on the DNA that you inherited from your parents. Phenotype has to do with observable characteristics, so hair color, skin color, height, build. Those are all examples of phenotype. So it could be that your phenotype shows that you have brown eyes. We know that why? Because we look at you and we can observe that you have brown colored eyes. However, it could be that your genotype says that you actually carry the allele for blue eyes. We don't see it because the brown eye gene is dominant or stronger, which is why your phenotype shows brown eyes. But technically, your genotype would still have that blue-eyed allele, which means that you could theoretically pass on that blue-eyed allele. Dominant versus recessive alleles. Most inheritable traits are controlled by more than just one gene, and in those cases it's what we call a polygenic trait. Other traits are controlled by one gene alone, and so your alleles on that gene can be dominant or recessive. In the example here on the slide, the capital A is the dominant allele for the flower color purple, and the recessive allele is the lowercase letter a for the flower color white. If you have a dominant allele, you will always express that in your phenotype. So whether you have two purple dominant alleles, which is capital A, meaning you got each of those from one of each from your parents, or if you received one dominant allele from one parent and one recessive allele from the other parent, either way, because of that dominant allele, that is the trait that will be expressed in your phenotype. For the recessive allele phenotype to be expressed, you would actually need to have two recessive alleles, and that's the only way that it would show. So when you have two recessive alleles, so two little a's in this case, that would be considered homozygous because it's two identical alleles, and in this case you would express the recessive trait of the white flower. If you have the homozygous trait of two dominant alleles, so two capital A's, then you would definitely express the phenotype of the purple flower. Now, in the case of your heterozygous gene, it means that you have two different alleles, meaning one parent gave you the dominant or the capital A, and one parent gave you the recessive or the lowercase a. But once again, because the dominant allele is present, that would mean that you would express the dominant trait in your phenotype. This is a picture of a Punnett square, and a Punnett square is a tool that we use to predict how genes will interact in the offspring that are produced. In this case, the capital B represents the dominant allele for a cleft chin, and the lowercase b represents the recessive allele. So since capital B, the cleft chin, is a dominant allele, and you see here that the mother's genotype includes she's heterozygous for the cleft chin, meaning she's got a capital B and a lowercase b. Dad, on the other hand, does not have a cleft chin, which means he is recessive. All he can pass on are lowercase b's because that's all he has. So in this example, you see mom is giving off either a capital B half of the time or a lowercase b half of the time, whereas dad is giving a lowercase b all of the time because that's all he has, which results in a 50% chance of the child having a cleft chin, because you see that 2 out of the 4 squares have the dominant allele, and a 50% chance of having no cleft chin, because you see that 50% of the squares, 2 of the 4 squares, are all lowercase. They're homozygous recessive. You have probably heard of the nature-nurture debate, nature referring to your biology or your genetics, and nurture referring to your environment. And the question is often asked, is it nature or is it nurture? And the somewhat snotty answer or response would be yes, because a lot of times it's a gene-environment interaction. The range of reaction asserts that our genes set the boundaries within which we can operate, and our environment interacts with the genes to determine where in that range we will fall. The genetic environmental correlation views one gene-environment interaction that asserts our genes affect our environment, and other environment influences the expression of our genes. Finally, epigenetics is the study of the gene-environment interactions, such as how the same genotype leads to different phenotypes. Now we're going to move on to neurons. So looking at the structure of a neuron, the function of the neuron is aided by specific aspects of its structure. Its semi-permeable membrane allows smaller molecules or molecules without an electrical charge to pass through it, while stopping larger or highly charged molecules. Incoming electrical signals from other neurons are received by the dendrites, which then send the signal down the axon. So here we have an image of a neuron or a nerve cell. This is a kind of prototypical or standard looking neuron. In the center, in this purpley pinkish color, we have the nucleus, which is not labeled here in the picture. Around the nucleus is the cell body or soma. Extending from the cell body or soma are these branchy looking things that are the dendrites. Of course, the external part of the soma would be the cell membrane. Now the soma is attached to the axon, which is a long, skinny tube essentially that goes down. And at the end of the axon, you see these terminal buttons, which is kind of they kind of look like dendrites, but they don't have the cell body there. I like to think of it as a used Q-tip, where one side is kind of more fluffy than the other. We also have myelin sheath, which are these, I guess, turquoisey blue, hot dog looking things that are going around the axon. The myelin sheath is actually a fatty tissue, and that fatty tissue helps speed up the signal that gets sent down the axon, which is called the action potential. So again, the action potential is a signal that travels down the axon, and the speed that it travels is only affected by the presence or absence of the myelin sheath. If you have myelin, the signal will travel faster. If you don't have myelin, the signal will travel more slowly. The synapse, or the synaptic gap, is quite literally the space between the terminal button of the sending neuron and the dendrite or soma of the receiving neuron. The action potential is the electrical signal that travels down the axon. The way it happens is that neurotransmitters from nearby neurons attach to the receptors on the dendrites or soma, causing the membrane potential to change. If there's depolarization, it means that the membrane potential becomes less negative, making the neuron more likely to fire. It's a state of excitation. Hyperpolarization is when the membrane potential becomes more negative, making the neuron less likely to fire. We call that inhibition. If the level of charge reaches the threshold of excitation, an action potential will occur. The ion charges open, causing a sodium ion to rush into the cell and the inside to become momentarily more positive. So when we talk about the threshold of excitation, that's the level of charge in the membrane that causes the neuron to be active, because otherwise it's just sitting there in a resting state. So once we hit that threshold of excitation, the action potential is released, and your action potential acts as an all or none principle. The incoming signal is either sufficient to reach the threshold of excitation or it is not. So I think I mentioned before that the speed of the action potential is only affected by the presence or absence of myelin sheath around the axon. If the myelin is present, the action potential will move more quickly. If the myelin is not present, the action potential will move more slowly. And so the speed of the action potential has absolutely nothing to do with the threshold of excitation. In other words, let's say a whole bunch of neurotransmitters land on this neuron, and it very quickly reaches that threshold of excitation. That action potential speed is, again, it's all in all. Once it's signaled, it's just going to travel. It doesn't matter whether there were a ton of neurotransmitters that helped reach that threshold of excitation or if you just barely met that threshold. You want to think of it like firing a paintball gun. It doesn't matter if you're an adult and you've got a whole bunch of finger strength and you can very easily pull that trigger to release the paintball. Or if you're a young kid who's trying to play with their big siblings' paintball gun, and so it takes them a whole lot of squeezing, possibly using like their whole little hand trying to get it to go, and they just barely have enough energy to reach that threshold of excitation. In other words, that ability to pull the trigger in which the paintball is released. And it doesn't matter if it was an easy press from the grown-up or the difficult time from the young kid, the speed of that paintball is not going to change. Once it's fired, it's fired. It doesn't matter how easily it was fired. A lot of people confuse it with the thought of an arrow, because that, of course, would depend on strength. So this is more like a paintball gun. Again, it's an all or none principle. Reuptake happens once an action potential has occurred, because now there's excess neurotransmitters in the synapse and they either drift away, are broken down, or reabsorbed. So reuptake involves moving a neurotransmitter from the synapse back into the axon terminal from which it was released. So if you think about it, so sometimes what happens is that you have such a great excess of neurotransmitters, and oddly they're not making their way across the synapse. Even though there's a bunch of them, they seem to just they've gotten to the habit of reuptake, being basically sucked back into the sending neuron. In situations like that, if it happens to be that the neurotransmitter is serotonin, and the serotonin isn't making it across the synapse, it can result in a person being depressed. And so certain medications, SSRIs, selective serotonin reuptake inhibitors, such as Prozac or Zoloft, or the old school ones, what happens is that once the serotonin is released, what happens is the medication basically blocks the openings so that the serotonin cannot get sucked back into or reuptaken taken by the sending neuron. And sure, some might still break down, but some might actually make it across the synaptic gap. I'm going to put up a video for you to watch. It's actually an old Zoloft commercial, but it actually shows you what happens when the medication is trying to help the neurotransmitter get across the synaptic gap. So neurotransmitters are chemical messengers of the nervous system. Different neurons are going to release different types of neurotransmitters that have many different functions. I'm looking at the biological perspective. They look at certain disorders like depression or schizophrenia, and they're associated with imbalances in one or more neurotransmitter systems. So it has to do with, neurotransmitters not properly getting across the synaptic gap, or maybe too much coming across the synapse. The neurotransmitter of acetylcholine aids muscle action and memory. Beta endorphin has to do with pain and pleasure. You've probably heard of endorphins. Dopamine has to do with mood, sleep, and learning. Norepinephrine has to do with heart, intestines, and alertness. And serotonin has to do with mood and sleep. We oftentimes refer to the serotonin as the happy neurotransmitter. Psychotropic medication are drugs that treat psychiatric symptoms by restoring neurotransmitter balance. We have agonists and antagonists. An agonist is a drug that mimics or strengthens the effect of the neurotransmitter, whereas the antagonist is a drug that blocks or impedes the normal activity of a given neurotransmitter. And so physicians prescribe agonists and antagonists to correct neurotransmitter imbalances. For example, Parkinson's disease is associated with low levels of dopamine. So dopamine agonists are often prescribed as one form of treatment. On the other hand, schizophrenia is associated with too much dopamine. So many antipsychotic drugs, therefore, are dopamine antagonists. The way that neurotransmitters work is that it's like a lock and key mechanism. Now, I want to point out this is not literally what it looks like. This is a visual to help you understand conceptually what's going on with the neurotransmitters. So neurotransmitters bind to the receptors of the receiving neuron in this key lock mechanism. So this turquoisey blue would be the neurotransmitter molecule. And this kind of orangey red is the receptor site on the receiving neuron. And you'll see here that the receiving neuron has a square, triangle space. And so the correct neurotransmitter to land would also need to have a square, triangle. And then it would land, and if enough land, then we could possibly meet threshold and hit the excitatory state. Once again, an agonist is similar enough to the neurotransmitter to mimic it. In our pretend example here, our pretend model, the receptor site has a space for a square, triangle. The agonist landing is square, and then flat. It doesn't have the triangle there. However, it's similar enough to the neurotransmitter to be able to land and actually activate the neurotransmitter. So if enough of these agonists land, it will trick the trick the neuron into thinking that it should be in an excitatory state. Morphine is an example of an agonist. If you have ever been in like a car accident or gone to the ER and needed pain medication, what happens is that there's just not enough endorphins to help you block all the pain that you're having, so they will inject you with morphine, which then floats around, lands on those receptor sites, and tricks your body into thinking that there are enough endorphins to make the pain go away. An antagonist, other than being a bad guy in a story, in this case is a molecule that inhibits. In other words, it's similar enough to be able to land in the receptor site, but it's not similar enough to actually trigger the neuron into thinking that there is something there, that it needs to be in an excitatory state. And so basically what happens is that as it sits here, it blocks the actual neurotransmitter from landing. So the example here is about acetylcholine. I'm going to give you a slightly different one. If you've ever heard of Botox, Botox is actually botulin poison. It's a botulin poison from pigs. And what it does is that it is an antagonist to acetylcholine. Acetylcholine, if you remember the earlier slide, helps with movement. And so when you inject this antagonist, you can't move the muscles in your face. And when you can't move the muscles in your face, then you don't see the wrinkles. And so when people are going for Botox injections to make themselves look prettier or less wrinkly, they are willingly injecting a poison that, if it gets into their bloodstream, could actually kill them, all for the sake of having their face not move so that the acetylcholine can't land, and that's why it's not moving and causing any wrinkles. Now we're going to move on to the nervous system. Just to start off, our nervous system divides into two major parts: the central nervous system, which is essentially the brain and the spinal cord, and the peripheral nervous system, which is just about everything else in the nervous system other than the brain and the spinal cord. So the peripheral nervous system breaks into two parts as well: the somatic or skeletal nervous system and the autonomic nervous system. Your somatic nervous system relays sensory and motor information to and from the central nervous system, and your autonomic nervous system basically controls all of your internal organs and glands. The autonomic nervous system can then be divided into two parts as well: the sympathetic and parasympathetic nervous systems. Your sympathetic nervous system has to do with stress-related activities and functions. It prepares us for fight or flight, or actually I've heard it as fight, flight, or faint, because some people just pass out. But generally speaking, if you're walking down a street, you think you hear a gunshot, it was really just a car backfiring, but you don't know, so you get scared. And so your two main things are usually to be prepared to defend yourself, that's the fight, or you run, which I think is what most of us would do, which is the flight. And of course there are the ones who either freeze or faint, and they actually don't move either because they are probably a little too anxious to figure it out. Now the parasympathetic nervous system has to do with day-to-day operations of the body under relaxed conditions. And basically it's what when the sympathetic nervous system kicks in, the parasympathetic nervous system will then kick in afterwards, trying to get you to calm back down again. So it relaxes the body after fight or flight. They like to call this one rest and digest, because when you are in fight or flight and your sympathetic nervous system is activated, your digestion slows down. And so these two systems, sympathetic and parasympathetic nervous system, both parts of the autonomic nervous system, complement each other and maintain homeostasis, which is an equilibrium within the body. So just a little chart to help you understand how this breaks down. So the nervous system breaks into the central nervous system, which is the brain and the spinal cord, and the peripheral nervous system, which is the nerves to and from the central nervous system. The peripheral nervous system breaks into somatic or skeletal nervous system, and the autonomic nervous system. So somatic is anything that deals with touch, hearing, and then of course the muscles that move your body. Autonomic is for internal senses, like your gut, smooth muscles, things like that. And then autonomic breaks down into sympathetic and parasympathetic. Sympathetic is your emergency. Housekeeping for parasympathetic is just the idea that we're trying to calm you back down and get you back down to normal. This little picture shows you what happens when your sympathetic and parasympathetic nervous systems kick in. So normally we are in a state of rest, and so our parasympathetic nervous system is working, so our pupils will constrict. It stimulates salivation, so if you see food that you like, you might start to drool. It keeps your heart rate down. It constricts the bronchi, the airways in your lungs. It stimulates digestion, and it causes your bladder to contract, which means that if you need to pee, you can hold it in. Your sympathetic nervous system, it kicks in because of a fight or flight response. Your pupils are going to dilate so that you can try to see as much of everything that's out there. You're going to stop drooling because right now digestion is not important. Your heart rate will increase so that your blood is pumping, and it also dilates the bronchi so that more oxygen is getting in, so you've got wonderful oxygenated blood flowing through your body to help you with that fight or that flight. Again, digestion has been stopped, and it stops contraction of your bladder. So there are some people who, when they get really, really nervous, their bladder releases and they can actually urinate all over themselves. So the brain is composed of billions of interconnected neurons and glia. Those are both kinds of brain cells. Your brain is bilateral, meaning it has two sides. We'll talk about that in just a moment. And it can be separated into distinct lobes, in other words, sections, but all of these sections interact with each other. You also have your spinal cord, which delivers messages to and from the brain. It has its own system of reflexes. So, when you go to the doctor and they hit you just below the kneecap and your leg kicks out and you can't control it, that's a reflex. So basically, there's a signal there that tells your foot to, or your leg to jerk out right away, just because of the fact that it got hit just there below the kneecap. Another reflex that you have that you may or may not have noticed, hopefully you haven't, is if you touch something really hot, your hand will jerk back for a second, and it'll jerk back, and then like a moment later you'll think, ow, that hurts. Why? Because the signal to jerk back is just between the spine and your hand, so that's a very close section, whereas for you to say, ow, that hurts, the signal has to travel all the way up to your brain and then tell you that it hurts. So that's why you have that there. There are two halves or two hemispheres of your brain, the left hemisphere and the right hemisphere. The concept of lateralization is the idea that each hemisphere of the brain is associated with specialized functions. Generally speaking, the left hemisphere controls the right side of the body and the right hemisphere controls the left side of the body. So if you are right-handed, you're controlled by the left hemisphere of your brain. If you're left-handed, you're controlled by the right hemisphere of your brain. When my kid was younger, she used to do this really cool thing. She's left-handed now, but at the time she was showing, she had better fine motor control with her left hand, that's what we would say, but she was somewhat both-handed. So she could actually start to color a page with one hand and then get to the top of the page, and then she would switch hands and color with the right hand for the rest of the page. And then when she was in kindergarten, one of the teachers saw her doing that and said, stop playing around and write with your right hand like you're supposed to. Now, of course, like we said, she'd been favoring her left hand, so when she told us a story, which she didn't think was anything wrong, she was just like, remember that cool thing I do when I color? Well, the teacher said this today. And so needless to say, her two PhD psychology parents wrote a letter explaining the concept of lateralization and how it's not set until you're like 6 years old, and so that the teacher should not be telling her to write with her right hand like she was supposed to. Now the corpus callosum is a thick band of neurons that connect the left and right hemispheres of the brain. And so that's how information gets from one side to the other. And you see here in the different pictures, just different views of the brain and the corpus callosum. The brain breaks into three major areas: the forebrain, the midbrain, and the hindbrain. The forebrain is most of the brain. That's part of what we think about when we think of like the brain doing higher functions. It contains the cerebral cortex, which is the higher level processes, the thalamus, which is sensory relay, the hypothalamus, which is homeostasis, the pituitary gland, the master gland of the endocrine system, and the limbic system, which has to do with emotion and memory. Now, the cerebral cortex is really just the surface of the brain. You can almost think of it like as the outermost layer of the brain. And it is associated with our highest mental capabilities like consciousness, thought, emotion, reasoning, language, and memory. The cerebral cortex can be broken up into 4 lobes. The frontal brain, which is the front of the brain just behind the forehead. The parietal lobe, which is kind of the top back of the brain. The occipital lobe, which is the base of your brain just kind of opposite your eyes. And then underneath your temples is the temporal lobe. The frontal lobe has to do with executive functioning, so planning, organization, judgment, etc. Also motor control, emotion, and language. So it controls the motor cortex, the prefrontal cortex, and what's called Broca's area. So the motor cortex is a strip of the cortex involved in planning and coordination of movement. And it's just basically the back part of the frontal lobe just before the parietal lobe. Your prefrontal cortex is responsible for higher level cognitive functioning, and it is not fully developed until you are 25 if you are male or 27 or 28 if you are female. So when teenagers and young adults are making decisions, are oftentimes making decisions when their brains are not fully developed yet. Now, Broca's area is a part of the left hemisphere in the frontal lobe that has to do with language production. And so damage to Broca's area will lead to difficulties producing language. And so the chart here explains about frontal lobe damage with Phineas Gage, who was a railroad foreman, and an accident caused an iron to penetrate through his skull and frontal lobe. And so after the accident, people noticed changes in his personality. So before the accident, he was really well-mannered and soft-spoken. After the accident, he started behaving in odd and inappropriate ways. And these changes were consistent with loss of impulse control, a function of the frontal lobe. If you've ever watched reruns of The Golden Girls, Sophia is supposed to have had a stroke that affected her frontal lobe, which is why she will say things like Rose is stupid and Blanche is a slut, because apparently the part of her brain that is able to filter things was damaged. Oh, I'm also going to upload a video or do a YouTube link to a video to show someone who had damage in Broca's area. And here's just a little slide about Phineas Gage. Now, the parietal lobe is involved in processing various sensory and perceptual information. It contains the primary somatosensory cortex, oftentimes just called the sensory cortex. And it's essential for processing sensory information from across the body like touch, temperature, and pain. And it seems to be organized topographically. So you can see here in the chart how it's organized. Now, the temporal lobe is located on each side of the head near the temples. It contains the auditory cortex, which is the part of the cortex in the temporal lobe that's responsible for the processing of auditory information. So while the somatosensory cortex in the parietal lobe does deal with most of the senses, the hearing tends to go through the temporal lobe. And so Wernicke's area is important for speech comprehension, and damage to this area results in difficulty understanding language. And I'll have a link for a video for this too. For this slide, I just want to point out that Broca's area is in the left hemisphere. So Broca's is in the Frontal lobe, Wernicke's is in the temporal lobe, but both sides, they both are on the left side or in the left hemisphere. So your occipital lobe, again, is at the back of the brain, opposite the eyes, kind of just above the brain stem. It's associated with visual processing. It contains the primary visual cortex, which is responsible for interpreting incoming visual information. I think after that, some stuff does get processed through the parietal lobe and the somatosensory cortex. The occipital lobe is organized retinotopically. I can barely say that word, retinotopically. Which means that it's mapped according to visual information from the retina to the neurons in the brain. The thalamus, which is still part of the forebrain, serves as the relay center of the brain where most senses, except for smell, are routed before being directed to other areas of the brain for processing. The limbic system has to do with emotion and memory. It's made up of a bunch of different structures. The most important ones are the amygdala, which is involved in our experience of emotion and tying emotional meanings to our memories. It also helps us process fear. The hippocampus, which is the structure associated with learning and memory, in particular spatial memory. And then the hypothalamus, which regulates homeostasis, which has to do with keeping your body temperature, where it's supposed to be, and your appetite and your blood pressure. Now, the midbrain has the reticular formation, which is important in regulating the sleep-wake cycle, arousal, alertness, and motor activity. Then there's the substantia nigra, where dopamine is produced, involved in control of movement. And then there's the ventral tegmental area, the VTA, where dopamine is produced, associated with mood. And we know that there's just degeneration of the substantia nigra and the VTA in Parkinson's disease. The hindbrain is probably the oldest part of the brain. It contains the medulla, the pons, and the cerebellum. The medulla controls automated processes like breathing, blood pressure, and heart rate, which is why, generally speaking, you don't have to think about doing those things. Then there's the pons that connects the brain and the spinal cord, involved in regulating brain activity during sleep. And the cerebellum, which controls our balance, coordination, movement, and motor skills. And it's thought to be important in processing certain kinds of memories. These three structures combined are known as the brainstem. These days we have amazing technology to do brain imaging. There's the CT or CAT scan and the PET scan that involve radiation. There's the MRI and the fMRI that has to do with magnetic fields, and then there's the EEG, which has to do with electrical activity. The CAT scan or CT scan stands for computerized tomography. It involves X-rays, and it creates an image through X-rays passing through varied densities within the brain. And so a CT scan can be used to show brain tumors or lesions, meaning like spaces in the brain. The PET scan, or positron emission tomography scan, is helpful for showing activity in different parts of the brain. It involves injecting someone with a mildly radioactive substance and monitoring changes in blood flow to different regions of the brain. The PET scan is helpful for showing activity in different parts of the brain. It literally lights up in colors with the scan. The MRI, or magnetic resonance imaging, uses magnetic fields to produce a picture of the tissue being imaged. And then the functional magnetic resonance imaging, or fMRI, is an MRI that shows changes in metabolic activity over time. MRIs don't have to be done on the brain, they can be done in other parts of your body as well, and they are completely noisy, very, very loud as the magnets are being used. An EEG is an electroencephalography, and it's involved with reading electrical activity of the brain via electrodes on the scalp. And so either they put a cap on you or they'll stick the electrodes on directly, and then they are measuring your brain activity as you're doing different tasks. Now, your endocrine system is a series of glands that produce hormones to regulate many body functions. The hypothalamus links the nervous system and endocrine system by controlling the pituitary gland. Again, the pituitary serves as the master gland, controlling the secretions of all the other glands. It's located in your brain. And you've got your thyroid, which is a little bit lower down, kind of around your neck, which secretes thyroxine, which regulates growth, metabolism, and appetite. Then you have your adrenal glands, which are on top of your kidneys, that secrete hormones involved in the stress response. So when you go into fight or flight, that has to do with the adrenal gland. And you've got your gonad, which is the secretes sex hormones. So for women, they're in the ovaries. For men, they're in the testes. And it's important for successful reproduction. It also regulates sexual motivation and behavior. And then you've got your pancreas, which secretes hormones that regulate your blood sugar. If your nervous system is like sending email, meaning it can send, you can send a message today or right now, and just a few seconds later, your friend all the way over in, let's say, Norway could have the message. Your endocrine system is kind of like snail mail or regular, stick a letter at the post office mail. The information will also get there, but it travels a lot more slowly. And here is just a slide that shows you the different neurotransmitters and how they affect your brain.